example of GCPR signaling involving epinephrine. Epinephrine binds to a beta-adrenergic receptor on the cellular membrane, a type of G protein coupled receptor on liver or muscle cells. This changes the receptor, activating an associated G protein by causing the G alpha subunit to exchange GDP for GTP. The activated G alpha subunit then stimulates adenylate cyclase, causing it to convert ATP into cyclic AMP. Increased CAMP levels activate protein kinase, which phosphorylates and activates glycogen phosphorylase while inhibiting glycogen synthase. Glycogen phosphorylase converts glycogen into glucose, providing a big energy source during the fight or flight response. The next signaling example is an ion channel. When activated by stimuli such as capsaicin, TRPV1 opens to allow calcium ions to enter the cell. The increase of calcium activates the enzyme calcineurin, which dephosphorylates the transcription factor NFAT2. Once dephosphorylated, the NFAT2 moves into the nucleus where it prevents the transcription of ATF3, a protein that blocks the activity of the cell cycle regulator DOG. Without ATF3, DOG induces apoptosis and prevents cell division.
The RTK, which is also known as receptor tyrosine kinase, cell communication is a process by which cells receive signals from their environment. In this case, it is epidermal growth factors, which is known as EGF, primarily through the binding of signaling molecules, ligands, like growth factors to receptors on the cell surface. This binding activates the RTK from the EGF receptor, triggering a conformal change that allows the intracellular part of the receptor to add phosphate groups, which is known as phosphorylation, to tyrosine residues within the receptor itself. This phosphorylation creates docking sites for other proteins. In our model, this is shown to be the MAPK kinases, initiating a cascade of intracellular signals that regulate critical cellular processes such as growth, survival, metabolism, and differentiation. This will change the MAPK kinase, which transforms the MEK molecule to an MAP kinase through phosphorylation. The signal is eventually turned off through mechanisms like dephosphorylation, which ensures that the process is tightly regulated. RTK signaling is essential for normal cell function, and the phosphorylation of cytolytic targets promotes growth and division. Intracellular receptors are proteins inside the help that help the cell to respond to signals from the outside, but unlike receptors on the cell surface, they don't need a signal to enter the cell membrane. Instead, these receptors are activated by small molecules, and in our model, this is shown by aldosterone, which can pass directly through the capillary membrane. Once inside the interstitial fluid, the hormone or signal molecule binds to the mineralocorticoid receptor, which then changes shape and acts like a switch, turning on specific cellular processes. This often involves the receptor moving to the cell's nucleus, where it can directly affect the DNA and control gene expression, essentially telling the cell to make certain proteins or perform specific functions. This is the site of transcription and translation of the epithelial sodium chloride channel and the NAK ATPase pump. Then there is the transfer of sodium from the tuberolumin to the ductal cell, which in this type of signaling is crucial for regulating long-term processes like growth, metabolism, and reproduction. And this is involved in things like how our bodies respond to stress or control energy balance. Next, let's look at DNA replication. DNA replication is essential in creating new skin cells, as it makes sure each cell has an exact copy of the genetic blueprint. Helicase begins this process by unwinding the double helix, separating the two strands to create a replication fork. Each strand serves as a template for building a new complementary strand. Before new DNA strands can form, an enzyme called primase lays down a short RNA primer on each template strand. This primer provides a starting point for DNA polymerase, which can only add new nucleotides in the 5' to 3' direction. Because of this replication of one strand, the leading strand is continuous, while the other strand, the lagging strand, is built in short segments called Okazaki fragments. Another enzyme, DNA ligase, joins these fragments together. This process enables basal cells in the skin's lower layers to divide and generate new cells. These cells can move upward to replace dead ones and heal wounds.
Next, we'll talk about mitosis. Mitosis is a process by which a cell divides to create two identical daughter cells. In the case of skin cell growth, mitosis is essential for replacing dead or damaged cells. The first stage in this process is prophase. Here, the chromosomes condense and become visible. The nuclear membrane begins to break down and structures called spindle fibers form, extending from opposite ends of the cell. In metaphase, the chromosomes align at the center of the cell, attached to the spindle fibers. During anaphase, the sister chromatids are pulled apart toward opposite ends of the cell. In telophase, the chromatids reach the poles and new nuclear membranes form around each set of chromosomes, creating two distinct nuclei. Finally, in cytokinesis, the cytoplasm divides and the cell pinches in two, creating two daughter cells, each with an identical set of chromosomes. This process is crucial for skin cell growth, as new cells are formed to replace old, dead skin cells.